Hello, dear guys. So here we continue our set of lectures about immunity, immunology, and previously we stopped on the topic on the part of MHC. What is MHC and HLA? So MHC and HLA is actually similar. So I remind you that major histocompatibility complex or main histocompatibility complex. There are two types of them. We have MHC1 and MHC2. So MHC1 is present almost on every, so not almost, but on every living cell. Every living cell possesses their own MHC1. So in this, and it depends on genetic code. So which HLA or which MHC is present. So Muhammad Faisal absent. Say Muhammad Faisal that he is absent, and Han Junaid Alam also absent. So and HLI, and we have MHC1 and MHC2. MHC1 is a major histocompatibility complex present all over the cells, in every cell of the body. But MHC2 is present only in cells of that show. So they are known as. It interrupts me and you. So say them if they will start to write me in WhatsApp. So I'll put 2 mark for a practical class to them. If they'll continue joining, I'll put 2 mark in practical class because really it bothers. Okay, and MHC2 is a molecule which is expressed so on antigen presenting cells. There are a few cells, antigen presenting cells, which are present. They are monocytes or macrophages, and also they are dendritic cells. And we will discuss about them a little later. So MHC2 is present there. So and they present the antigen to other cells, especially to T lymphocytes. So we'll discuss what is MHC action a little later, but now let's talk about B lymphocytes. So B lymphocytes are derived from bone marrow. The bone marrow derived, and this word B is correlated, respond to the Fabricius bursa. So bursa cells, it's present in birds, but in males we have analogous to bursa lymphocytes. They are present in bone marrow. So Haider Muhammad Safir 2 mark. B cells make up 10 to 20% of circulating peripheral lymphocyte population. So and on the surface of B lymphocytes, they recognize the antigen with the help of immunoglobulin B. So especially, so you remember that immunoglobulin, so B cell immunoglobulin M. So you remember them from immunity. So there, this class of antibodies is it's a fast reacting antibody, so which is present on the surface of B cells. Okay, they are present unlike others. So this immunoglobulin M is attached to the cell membrane of the B cell, and along with other signaling molecules, it forms the BCR. It's known as B cell receptor. Okay, T cell recognize only MHC associated peptides. And it's different, but whereas the B cells can recognize and respond to many more chemical structures. So they can interact not only through the MHC, but they can interact with soluble or cell associated proteins, lipids, polysaccharides, nucleic acids, and small chemicals without a requirement of MHC. So you see, MHC is not required for B lymphocyte. It uses it, but it is not required so all the time. So here, unlike this, T cell receptors, each antibody has a unique amino acid sequence. So this antibody, so like T cell receptors, they are like antibodies, unique sequence. Yeah, we discussed before, and antibody is also unique sequence of amino acids. You remember that it's related to immunoglobulin genes. We have specific Ig genes in our B cells, so in every cell actually, but they are active in B cells. and due to rearrangement of this Ig genes, because of different arrangement of this genes, so genes can change their locations, they're drifting from one side to another, and that's create so absolutely non sorry, it creates absolutely not absolute specific antibody which were never present in our body, so a specific protein which is new for us. So some are signaling molecules attached to the BCR. Another example is CD21. CD21 is a special cell marker, so which actually is attached with IgM, so it's composed of immunoglobulin beta and Ig alpha. So they're attaching with IgM, and when they bind with IgM, so this CD21 attach the antigens which are attached with complement. You remember that complement, especially C3b, so it when it's attached with complement system, so these particles of antigen, they're analyzed by B lymphocytes. Okay. And actually, you remember that B lymphocyte is responsible for immunoglobulin synthesis. So we have different classes of immunoglobulins. They include the immunoglobulin G, immunoglobulin M, immunoglobulin E, immunoglobulin D, and immunoglobulin A. All of them are secreted outside, and about features of them we'll discuss a little later. Then let's talk about natural killer cells. Natural killer cells. So 1 minute. So natural killer cells, they are known as innate lymphoid cells. NK cells are lymphocytes that arise from the same common lymphoid progenitor that gives rise to T lymphocytes and B lymphocytes. You remember it from the topic of blood formation, yeah? But NK cells are they're innate. They facilitate the innate immunity. So they are functional without prior activation and do not express highly variable and clonally distributed receptors for antigens. So that's, you see, NK cells, they don't release specific antigens on the surface, and so Asra Khalid also 2 mark. So say here that she got 2 mark. After Wakas 2 mark in practical class. Please, group leaders, once again, ask your group mates not joining during the lecture. Okay, so now ask them, so order them, don't join during the lecture. So if they didn't join in proper times, then they have absentee and 2 mark. So if they try to join during the lecture, I give them 2 mark. Okay, so that's why attend every class at proper time, not later, or don't join at all. Okay, natural killer. Natural killer cells have 2 types of receptors. They are inhibitory and activating receptors. Inhibitory and activating receptors. So inhibitory receptors, they can recognize the MHC1 molecules, and the MHC1. So as I remember, so they are present in all cells. But activating receptors, they recognize molecules that are expressed on or upregulated on stressed or infected cells. So you see, activating receptors, Muhammad Hassan, 2 mark, group 2. Group leaders, have you said to your group mates that they shouldn't join now? Should not join now. Okay. We have natural killer cells and innate lymphoid cells. So okay, we discussed we have inhibitory receptors and activating receptors. Inhibitory receptors are which recognize self MHC molecules and prevent the autoimmune aggression to them. You see, these inhibitory receptors prevent the autoimmune aggression. But activating receptors, they are acting on changed cells. So they're, for example, especially viral infections or malignantly changed cells. So Okay, and we have so they have reduced expression of class 1 MHC molecules and increased expression of proteins that engage activating receptors. So NK cells, you see, they are activated, and the infected or stressed cells are killed and eliminated. NK cells also secrete cytokines such as interferon gamma. So, and you remember from the topic of inflammation that interferon gamma activates the classical pathway of macrophages. Classical pathway of macrophages, which cause the destruction of microbes or virally infected cells. So that's the main activity of this NK cell. So you see, this NK cells function is clear. And we have innate lymphoid cells. It's a very rare case of leukocytes. So lymphocytes, there are populations of lymphocytes that lack TCR, so but produce cytokines similar to those that made by T cells. You see, toll-like receptor, T cell receptors. They are producing cytokines after their activation, but without TCR, they are releasing this. They produce interferon gamma, interleukin 5, interleukin 17, cytokines that are characteristic of T helper 1, T helper 2, T helper 17 subsets of T cells. Later we will describe it, of course. So NK cells are related to group of 1 ILCs. Based on their production of interferon gamma, a cytokine also made by Th1 cells. So this interferon gamma is produced by T helper 1 cell. Innate lymphoid cells mostly reside in tissues. They are thought to provide early defense against infections in the tissues before T cells are activated and can migrate into tissues. So there, it's present in in the lamina propria of organs, yeah, for example, and where T cells are not charged yet, they're not attracted to the site, and they just act instead of T cells. To activize so for example, by production of interferon gamma, it activates the macrophages. By secretion of interleukin 5 or interleukin 17, it activates different cells like eosinophils also. Okay, that's and actually later this innate lymphoid cells won't be discussed, so because it's not so important. Then let's talk about antigen presenting cells. We already said about antigen presenting cells. They are very important cells of immunity. They just produce on the surface of antigen presented cells we have MHC1 and MHC2, major histocompatibility complex 1 and major histocompatibility complex 2, which I remind you that function of this histocompatibility complexes is to reveal the antigen to the surface of the cell and to present it to T lymphocytes predominantly. Okay, the immune system contains several cell types that are specialized to capture antigens and display this to lymphocytes. Foremost among these antigen-presenting cells are dendritic cells, and the major cells of displaying protein antigens to naive T cells, and also macrophages, so they are not shown in this description. And they are present this antigen in different stages of immune responses. So let's talk about dendritic cells. So in dendritic cells, they are most important antigen-presented cell for initiation T cell responses against protein antigens. So if we have protein antigen in the bacteria, so we need to present it. So we need to give opportunity to our T cells and B cells, so especially T cells, to recognize it and produce T cell receptor against it. If we talk about T lymphocytes. They are present, so dendritic cells, they are present in different locations. For example, under the skin, they form the Langerhans cells. Might be in histology discussed. So the Langerhans cells, Langerhans cells. Don't confuse with islets of Langerhans. They are not similar. It's absolutely different cells, but same scientists just found them. Okay. and they're located at the right place to capture antigens under epithelia, the common site of entry of microbes and foreign antigens, and in the interstitia of all tissues where antigens may be produced. So this you understand that they make a first capturing of these antigens. Um, so 1 minute. Okay, let's continue. So dendritic cells express many receptors for capturing and responding to microbes and other antigens, including TLR, toll-like receptors, and C-type lectin receptors. So I remind you, C-type lectin receptors, they play a major role in activation of lectin pathway of complement system. In response to microbes, dendritic cells are recruited to the T cell zones of lymphoid organs, where they are ideally positioned to present antigens to T cells. So you remember we have lymphoid organs. We'll discuss lymphoid organs a little later, but you already know that it's a collection of lymphocytes in one area. So lymphoid organ is a collection of lymphocytes, and these dendritic cells, they have this appearance. So no. Dendritic cell is known dendritic because of this appearance. It's a star-shaped, stellar-shaped cell with a lot of outgrowth of the cytoplasm, and these outgrowths are present between these T cells, and then the T cells can teach on it. So can have attempts to destroy these antigens, and are teaching there to find this antigen and capture it. So that's main function of dendritic cells. Dendritic cells express high levels of MHC and other molecules needed for antigen presentation and activation of T cells, as I said it before. So we have two types of dendritic cells. There are plasmacytoid cells, because of their resemblance to plasma cells. They're present in the blood and lymphoid organs, and major sources of the antiviral cytokine type 1 interferon. It's a produced response to many viruses, so that's main function of the production of interferon 1, type 1. And the second type is known as follicular dendritic cell. So it is present in the follicles of lymphoid organs in germinal centers. So I'll remind you, germinal centers is the site of proliferation of lymphocytes, predominantly B lymphocytes, and they are also present there for that function. Okay, these cells bear Fc receptors for immunoglobulin G and receptors for C3b, and can trap antigen bound to antibodies or complement proteins. These cells display antigens to B lymphocytes in lymphoid follicles and promote antibody responses, but are not involved in capturing antigens for display to T cells. So you see, so it seems like similar cell, but it gives a little different function. So okay, they are like they have Fc receptors. They bear Fc receptors for immunoglobulin G. Okay, so only which are already bind with antibodies. Okay, 1 minute. Okay, and we have other antigen presenting cell which possess less important function, so less antigen presenting function, but more phagocytic function. They are macrophages. And functional macrophages has already discussed it. We possess a lot of different functions, but they can ingest microbes and other particulate antigens and display peptides for recognition. So they also have the same HC molecules, and this. So T cells in turn activate the macrophages to kill the microbes, the central reaction of cell-mediated immunity. So you see, macrophages, they work in two ways. So it's antigen-presented cells which activate the T lymphocytes, but activated T lymphocytes promote the synthesis interferon gamma, which causes the activation of macrophages. So you see, they're mutually depend on each other, and that helps to maintain the macrophage activity. B cells present peptides to helper T cells and receive signals that stimulate antibody responses to protein antigens, critical steps in humoral immune responses. Okay, let's talk about lymphoid tissues. So we have so it's from histology, you should have known it. So with this lymphoid tissues, we have central lymphoid organs and peripheral lymphoid organs. Central lymphoid organs, they are like T lymphocytes, so thymus and bone marrow. So in which T lymphocytes and B lymphocytes get mature and became competent. And peripheral secondary lymphoid organs, which include diffuse lymphatic tissue, also lymph nodes, lymph nodules, and spleen. So they are peripheral lymphoid organs. So briefly, let's talk about peripheral lymphoid organs. So they are organized to concentrate antigen-presenting cells, and lymphocytes in a way that optimize interactions among the cells and the development of adaptive immune responses. Most of body lymphocytes are collected there. So you see, it's a specific site where lymphocytes are very closely packed with antigen presenting cells. It's like I can compare this with the universities. Yeah, it's the universities where we teach the specific cells in one area. We have to collect it in one place and then teach you how to act on these antigens. And that's a perfect organization is peripheral lymphoid organs. Okay, let's start. Lymph nodes. So, by definition, it's encapsulated, highly organized collections of lymphoid cells and innate immune cells that are located along lymphatic channels throughout the body. So, as lymph passes through lymph nodes, resident APC are able to sample antigens and that are carried to the node in lymph derived from the interstitial fluids of tissues. So, look, in easy words, what is a lymph? Lymph is the interstitial fluid. Yeah, interstitial fluid, and interstitial fluid is present blood vessel, so under the endothelium. Under the epithelium, sorry. So epithelium is present between outer environment and inner environment. Outer environment is full of bacteria. So and of course under these epithelial cells, we have loose connective tissue. And this loose connective tissue possesses a lot of ground substance, a lot of liquid, a lot of compounds. Sakibula. So group number 5. Say to your group leader that he late and he interrupts me and he got bad mark. So and don't write in WhatsApp. So 1 minute. So let's talk about lymph nodes. I start to talk about lymph. Yeah, so lymph is interstitial fluid in loose connective tissue first, and in loose connective tissue, this loose connective tissue contains a lot of ground substance, and bacteria is appeared under the epithelial cells. They are present in loose connective tissue. They are present in liquid. And there, you remember from histology, in loose connective tissue, we have a lot of migratory cells like lymphocytes, neutrophils, macrophages, which are reacting on it, and they are producing. They can destroy this bacteria and can create antigens, and then all this fluid is collected inside the lymph nodes. And these antigen-presenting cells, like dendritic cells, they capture these antigens and place it on MHC on the surface, major histocompatibility complex. And by this feature, it can present it to T lymphocytes, which are present in lymph nodes. So that's the main idea of lymph nodes, why they should be collected and why they collect all the interstitial tissue. Because previously I remember, so when I was student and also as a student replied to me when I teach them histology, that they think that lymph is necessary only for regulation of liquid flow in our tissues. But main idea of lymph is to prevent antigens to enter into the general circulation without analysis of this by immunity cells. That's why all the liquid which it doesn't fit inside the blood, it's entered into the interstitial space, and from interstitial space it enters into the lymph nodes. Okay, and spleen. Spleen is a similar. It's also might be named as a very large lymph node, but spleen, unlike lymph node, it collects the blood. So all the blood which if bacteria or antigen is present inside the blood, so spleen will handle it. So in case of sepsis or septicemia or bacteremia, so this bacteria they somehow directly entered into the blood. So might be by injection, might be by impairment of lymph node function. But anyway, if antigen entered into the blood, they are analyzed by spleen. So sometimes they name it as largest lymph node in the body. Okay, that's how it looks like. Lymph node looks like. So this real image. So you see, lymph node is composed of follicles, and in these follicles you see in these follicles inside we have this white, so pale appeared cells. These pale appeared cells are known as germinal centers. Germinal centers. This germinal center is the site of cell maturation and cell growth. So I mean proliferation of cells. That's why it requires more cytoplasm. That's why nucleuses are much looser here, and that's why it's visible like paler region. And here lymphocytes are differentiated and grow in number. But between these follicles, so in follicles we predominantly have B lymphocytes. So main idea, B lymphocytes are present in follicles. But between follicles, we have predominantly interstitial lymphocytes. They are known as T lymphocytes. And these T lymphocytes are present between these follicles. So, and they also require the same feature, you see. But B lymphocytes are more organized in follicles, but T lymphocytes are present diffusely between them. So, and here inside the lymph node, we have this feature. You see dendritic cells which capture the antigen, and with this antigen present on MHC surface, it enters with the fluid into the lymphatic vessel, and from the lymphatic vessel enters into the lymph node. And then lymph node, so with antigen on the surface of it, so it meets the naive T cell, which captured from artery, and it teaches the naive cell to differentiate this antigen, which dendritic cell found in interstitial space. So we have T cell zone, B cell zone. So in histology you discuss it well. Okay. Okay, and the cutaneous and mucosal lymphoid systems are located under the epithelia of the skin and the gastrointestinal and respiratory tracts, respectively. So these cutaneous and mucosal lymphoid systems are known as diffuse lymphoid system and lymph nodules. They are present there. For example, the best example of them is tonsils, like pharyngeal tonsils, and also pierced patches in ileum of digestive tract. They are collections of lymph nodules. So that's like, so here you see, I said that here we have follicles, and these follicles are also named as lymph nodules. And if it's not collected in lymph node, if we see follicles but out of the lymph node, they are named as lymph nodules, and they are present in the digestive tract like pierced patches, and tonsils are very improved lymph nodules in oral cavity. Okay. Within the peripheral lymphoid organs, T lymphocytes and B lymphocytes are segregated into different regions. In lymph nodes, B cells are concentrated in discrete structures called follicles or nodules, located around the periphery or cortex of each node. So, in the middle, they contain germinal center, pale stained region, as I explained it before. The T lymphocytes are concentrated in the parafollicular cortex, interstitially between these follicles. The follicles contain the FDCs that are involved in the activation of B lymphocytes, B cells. It's follicular dendritic cells. You remember? We discussed it before. And the paracortex contains the DC, so just normal DC, so dendritic cells that present antigens to T lymphocytes. In the spleen, T lymphocytes are concentrated in periarterial lymphoid sheaths. You remember it from histology, surrounding small arterioles, and B cells reside in the follicles. So you see B lymphocytes are usually present in follicles, T lymphocytes are present interstitially. So lymphocytes constantly travel between tissues and home to particular sites. Okay, let's continue talk about it. So cytokines. We have different cytokines, messenger molecules of the immune system. Cytokines are secreted proteins that mediate immune and inflammatory reactions. These cytokines molecularly defined cytokines are called as interleukins, and we already heard about interleukin 1, interleukin 6, yeah, in cases when we discussed the fever and the protozoal fever. Okay, different cytokines contribute to specific types of immune responses. For example, innate immune responses cytokines are produced rapidly after microbes and other stimuli are encountered. So these cytokines like TNF, tumor necrosis factor, you remember it's produced by macrophages predominantly. Interleukin 1, also produced by activated macrophages. Interleukin 12, type 1 interferons, interferon gamma, and chemokines. So they are produced by lymphocytes predominantly. Their major sources are macrophages, dendritic cells, lymphocyte cells, and NK cells, as innate lymphocyte cells. But endothelial and epithelial cells also can produce them, so it's very much rare present. In adaptive immune responses, cytokines are produced principally by CD4 positive T lymphocytes activated by antigen and other signals and function to promote lymphocyte proliferation, differentiation, and to activate effector cells. So they are interleukin 2, interleukin 4, interleukin 5, interleukin 17, and interferon gamma. So all of them are produced by the cells, but also they promote the secretion, promote the differentiation and proliferation of the cells. So some cytokines serve mainly to limit and terminate immune responses. So this TGF. TGF, I remind you, so it's transforming growth factor, which is produced by macrophages. It's produced in a secondary alternative pathway of macrophage activation, and also interleukin 10, so which it expressed in chronic inflammation, you remember, and in these cases we just little depress the immunity in this area because we understand that we cannot handle it and we can depress it, or might be in a full resolution period, so interleukin 10 is also produced. So some cytokines stimulate hematopoiesis, and they're called colony stimulating factors because they stimulate formation of blood cells colonies from bone marrow progenitors. So you remember that granulocyte, monocyte colony stimulating factors, they promote the secretion of more neutrophils, eosinophils, monocytes. It's happened due to interleukins production. So interleukin, first of all, interleukin 1 in TNF, when it's present in the bone marrow, they cause a stimulation of this formation. And for example, in interleukin 7, it promotes the synthesis of T lymphocytes. Okay, that's we discussed. So what about time? So how lymphocyte activation and adaptive immune responses happened. So first of all, let's show it on diagram. Okay, so here we need some details. Microbes and other foreign antigens can enter the body virtually anywhere, and it's obviously impossible for lymphocytes of every specificity to patrol every possible portal of antigen entry. So that's okay. Let's skip it. Let's talk about. So you know it, and you can read it. So During the innate response, the microbe or adjuvant activates APCs, antigen-presenting cells, to express molecules called co-stimulators and to secrete cytokines that stimulate the proliferation and differentiation of T lymphocytes. So you see, when antigen-presenting cells are encountered with any kind of bacteria or antigen, co-stimulators, they activate the co-stimulators and factors of stimulation of T lymphocytes, for example, like B7 protein, another name CD80 or CD86. No need to remember them by heart. They are expressed on APCs and recognized by the CD28 receptor of naive T cells. You remember we discussed the CD28 receptor, so it's a receptor with which okay, we discussed it in first lecture of immune response. Okay, antigen and costimulatory molecules were produced during innate immunity response to microbes, function cooperatively to activate antigen-specific lymphocytes. Okay, okay, that's not important. So you see, cell-mediated immunity. So how cell-mediated immunity activated? How T lymphocytes are activated? Naive T lymphocytes. Naive, you understand, naive lymphocytes are which are never encountered with antigens. They are unable to kill no one. They are unable to kill anyone. But they are can be activated by antigen and co-stimulators with help of antigen presenting cells, and they can proliferate and differentiate into effector cells that migrate to any site where the antigen is present. So one of the earliest responses of CD4 positive helper T cells is secretion of cytokine interleukin-2 and expression of high affinity receptors for interleukin-2. So interleukin-2 is a growth factor that acts on these T lymphocytes and stimulates their proliferation, leading to an increase in the number of antigen-specific lymphocytes. Okay, I see that here we have a lot of information. I guess we will discuss it in next class. So uh, for today is it's enough. So lecture is over. So let's check attendance. Who's present now? And also here's a hype guild, so which is not shown in part. Okay. Thank you for attendance. See you next time. Goodbye.
Okay, sir. Thank you.